I'm sure that all of you have heard of the concept of cloning. What you may not realize is that cloning has been around since 1885. So it's not new technology. The technique, however, that we use today that's created a lot more buzz is very different. And to understand the value of the new way of cloning, you have to first go back to understand how the original type of cloning was done. So the first thing that you need to understand is that cells, when they start off, when they first come together, so let's say we have here sperm and the egg. When the first cell is formed via fertilization, we call that cell a zygote. And a zygote has two sets of information, two sets of DNA, one from the mother, one from the father, brought in through the sperm and the egg. And every single cell that forms from that zygote through cell division will have that same combined set of DNA that was brought together by the sperm and the egg. So the question you might have is, if you have this cell that exists in the zygote, how on earth does it actually specialize to become the different types of cells that you have in your body? What's interesting here is that, and we've talked about this before, despite the fact that a liver cell and a skin cell and a nerve cell and a muscle cell do very different things, they all have the same instructions. They all have the exact same DNA. However, a liver cell is going to read portions of the DNA that are not read by a skin cell, and a nerve cell will read a different section, and a muscle cell will read a different section. So while every single cell has a complete set of instructions to be any type of cell, once a cell has committed itself, once it's become specialized, then it can only read that part of the book, so to speak. This process of specialization, where certain parts of the DNA are turned off, is called differentiation. And once a cell has differentiated, there's no going back. It's a one-way street, in a sense. So that means if I were to pick off a skin cell from your body, and despite the fact that it has all the information to make every other cell in your body, if I were to put it on a petri dish and grow more of those cells, they would always and forever continue to be skin cells. So I'd have a big pile of skin cells, which is not obviously a clone of you as a human being. Now this is a pretty amazing thing to imagine here, because what's happening here is you could imagine that the areas that I'm bracketing off here, these would be areas of your DNA molecule that, say, a skin cell will not read. So those areas are off limits to a skin cell. A skin cell will only read this sequence right here and this one right here. And again, every skin cell that comes after, every generation that comes after, will only be able to read those red regions. Their X regions, or the green regions, will be off limits to that particular cell. Now this brings us back to this concept of the zygote. Now, as I said, the zygote consists of two sets of DNA, one from the mom and one from the dad, and we denote that by calling it a 2N cell, or a diploid cell, di meaning two, ploidy meaning information, so a diploid cell. And at this point, when the sperm and the egg come together to create the zygote, which will have a nucleus in it, so I'm just going to put our nucleus right there, this cell is completely undifferentiated. It has not yet specialized. And in fact, many generations after this, when the zygote divides into many smaller cells to look like this, all of these cells have not yet differentiated either. So while they are smaller in volume, they still have the exact same DNA, and the DNA hasn't been turned off in any sort of particular way. It's all available, and that means any one of these cells could give rise to any type of cell. When cells can still give rise to many different types of cells, we call them pluripotent. And pluripotent is actually a word that allows you to understand what it means. Pluri means many, potent means potential. So it has the potential to be many different cells. So every single one of the cells that you see in this bundle of cells are pluripotent. And in fact, when you have a bundle of cells, this pile of cells, which may be hard to fathom here, but it's actually a hollow ball of cells here. We call this a blastocyst. Okay? That formation is called a blastocyst. So when you're in a blastocyst, any one of the cells you have, you could pick off and it hasn't yet differentiated. Now, one thing that's really vital for you to understand here is that if we go back to our egg, if you recall, our egg only has one set of information. It's a monoploid, as opposed to the zygote, which is a diploid. And the same can be said for the sperm. The sperm is also a monoploid. The sperm, however, if you notice here, is much smaller than the egg, and in fact, the sperm is really pretty much useless except for the fact that it carries DNA. The egg, however, carries something that's really important. Not only does it carry DNA, it also carries something called factors. And these factors are a whole bunch of chemicals that essentially instruct the cell how to turn itself into a specialized set of cells. So one egg contains all those factors, and all those factors are available when the zygote forms to all the cells that will give rise to the blastocyst. So this blastocyst set of cells have access to all these factors. Now we still don't know exactly how these factors lead to the formation of the different types of cells. That's something that scientists are working on right now. But we do know that without those factors, you would never lose this pluripotency, that a cell, they would just become a blob of generic cells, and they would never develop into a full-fledged embryo, fetus, human being. So those factors are absolutely essential for the proper development of a human being. Now this has a very important implication. If every single one of these cells has a set of factors that will allow it to then later specialize properly and lead to the development of a human being, that means any one of these cells could give rise to another whatever organism. So if this is a human being, it could give rise to another organism. That's a really important thing because that's the concept that was understood and what led us to understand how to make the first clones way back in the late 1800s. So imagine we have this blastocyst, and this thing is embedded inside the uterus of a mother. So this is inside the womb of a human being. And if we had to, if we could, and in fact we certainly did have the ability, we went in and we culled out, we drew out this particular cell right here that was on the edge. And we took that single cell, which has factors as well as the DNA in its nucleus, and we implanted this into the womb of a surrogate mother. Oops, that's not how you spell surrogate. Now, this mother has absolutely no genetic connection to this cell that we see right here. Okay, so there's not going to be a relationship. All the surrogate mother does is provide a womb, which is the environment that will be used by this growing embryo to develop properly. But don't forget that this developing embryo, this egg not really this egg here, but this embryonic cell here has all the factors that it needs. And so what will happen here is that even though it has a low volume, that's not a big deal because the nutrients and such that come from the womb will allow this thing to divide and still grow into a full-sized cell. So that ultimately what you're going to have maybe a couple days later is another ball of cells. And that ball of cells are going to be identical to the original ball of cells that you see here that are hanging out inside the womb of the actual mother. All right? So if we let these things come to term, the one inside the womb of the actual mother and the other inside the womb of the surrogate mother, and if they come to term and both are born, then we're going to have twins, which is kind of an amazing thing to consider here. Because those factors will have the same impact on the DNA, and the DNA will lead to differentiation, proper differentiation associated with all the ball of cells, and ultimately will have completely identical twins. Now, one interesting thing to know, to confirm, the easiest way to confirm whether you have identical twins is to take a skin sample and swap it out. Now, this sounds a little bit cruel, but what you do is you take off a little chunk of skin, and I'm talking about the size of an eraser head, and from both of the organisms that you think are clones, and you swap them. In other words, you put the little disc of skin in the place of where the skin was removed from one of the babies, and put that piece that you just removed from one of the babies into the other open hole there. And as long as it takes, we know that the body doesn't reject something that thinks it's their own, part of their own body. And so our immune system is able to discern minute differences that exist between organisms. So if it doesn't reject it and it's able to take, the skin graft is essentially able to take, then you know that you're going to have full out clones of each other. So this seems like a pretty cool thing, right? So we've known how to do this. Now, we didn't do it with human beings in the 1800s. We did it with, I don't even remember what it was. I think it was some sort of sea urchin. I can't even remember. Anyway, and then, since then in the early 1900s, we did this embryo twinning, which is the process with a vertebrate, a salamander. And we understood that we were getting clones. We didn't quite understand what were the controls, like what were the nuclear controls that existed there. And then we took it a step further. Rather than simply taking an entire cell out, we take a cell, we would actually have a nucleus in it, but we would remove that nucleus. And we would then take another cell that had a nucleus. Oh, I want to make that black, hold on. That had a nucleus, and we would cut out that nucleus out of that cell. All right. Removing, we removed it from that cell, it no longer has those factors, right? So that's an important thing. And then we transferred this nucleus into a cell that did have the factors but was enucleated, didn't have a cell. And we were able to have the exact same situation happen here, where we would be able to create a clone of the organism once again. All right, the same deal that we had before. So it's just a slightly different thing. Instead of using an entire cell, we actually just did a nuclear transfer. Now, don't forget, these cells are not differentiated in any sort of way. Now, the reason why that's important is if you really want to think about how useful cloning would be, cloning is going to be useful if we have an adult form of something. It doesn't even have to be a human being. It could be any kind of organism. Maybe it's a cow that produces a huge amount of milk, more than most of the other cows. You want to clone that cow. You don't want to, you could certainly breed that cow with another cow, and maybe chances are you're going to have something that will be able to produce a lot of milk, but you can't know for certain if that's the case. The only way that you could know for certain that you'd be able to produce this super cow is to do a complete clone of the super cow. Keep in mind, though, what we're looking at here are cloning embryos. And when you clone embryos, you have no idea what the adult form of that organism is going to be. So you can't know that it's going to be really good at producing a lot of milk. What we want to do is take an adult and clone the adult because we know exactly what the adult's characteristics and traits and behaviors are. That's what we do in the next screencast when we talk about how we can clone adult forms of organisms.